All right. Hi, everyone. I am Lauren Louie. I'm a research scientist at Lawrence Berkeley National Lab, and I'm going to be talking about long read apps that were put into K-Base with John Mark Shandonia, Torben Nielsen, and then Paul Adams is the PI. I'll be talking about how to apply these apps for doing genome assembly with long read sequencing. And I'll also talk, I think it's really important to understand why it helps with genome assembly so you get the correct amount of sequencing data that you need for good assemblies. And as part of adding these apps into KBase, we actually ran a workshop in December, which I'll talk about. And we have some recordings and resources from that workshop as well that are available. And then I'll go through a K-base narrative of an example, and then Ben Allen will also talk about how you can publish genomes and other resources in K-base that you could apply to genomes. So here's an overview of the apps that we put in. So there's FiltLong, which helps you quality filter long read data. There's Unicycler, which is a hybrid assembler. And then because long reads are not as accurate as short reads, you can use something called polypolish to polish your assemblies. And then there is a long read only assembler called Fly that we also put in. So the motivation for putting these apps in is just because the technology is evolving. So if we, genome assembly apps are the most used apps in KBase. And it kind of makes sense because a lot of the things that we do are all sequence-based, microbes or plants or any organism, right? We use that as kind of a base for our knowledge of what we're doing in biology. And so since the technology is transitioning into long read sequencing, it makes sense to add these apps into KBase. And so another part of these types of apps is you do need computational resources. So if you can do the sequencing, but you don't have the computational resources, you can do it in KBase. So this was really also motivated by the work that I'm involved in with this Enigma science focus area. So we have a focus on trying to get as many isolates as possible to characterize the field site that we're working at. And so we developed a pipeline. We're sequencing. The goal is to sequence thousands of isolates, right? So if we can, then we can bring them into the lab, but then we still need to understand what their genomes are. We can track them through metagenomes. We can make mutant libraries out of them, but the genome is important. So what's the big deal of long reads? Basically, so because we have so much data in Enigma, I could actually make this graph of if you do, what happens if you actually increase the amount of coverage that you have of your genome. And so you can see here that after about 200x coverage, so that's the sequencing depth, right? You don't really improve your genome anymore. And that's because these are repeat limited. The reads aren't long enough to overcome the length of the genomic repeats, so you end up with fragmented assemblies. And so if you just add a little bit of long reads, which is what the genomes are for these triangles, these black triangles here, you can finish and circularize these microbial genomes. So the reason why long reads help can be illustrated in this assembly graph here. So modern assemblers create what are called assembly graphs. And so you can kind of think of these different bars as contigs, and these little edges here as how the contigs might be overlapping with each other, but there's ambiguity here. So this assembly graph actually represents two different possibilities. You can go around once, the blue and the pink here, or once with the blue and the green, or you can traverse it twice and get one big genome with 2 repeats in it. So if you actually have a long read that spans the repeat and it has both part of the blue and part of the green, then you know that it has to be the second choice. So that's how long reads help with assemblies. And actually, I'm not showing it here, but the long read assemblies also tend to be longer than the short read only assemblies. So there's some repeats that are getting collapsed or you're missing genes if you don't finish the assemblies. Another reason why we're so interested in these long read assemblies is because we can start seeing which genomes have plasmids. And so this is an example from one of the collaborators in Enigma. And so this one bacillus strain that's very resistant to heavy metals actually had 8 plasmids. And a lot of these plasmids had metal resistance genes. So this, doing the long read sequencing helped us see this part of the genome. After we put in all of the apps in the KBase, it really became apparent to me that there was a very big connection between what was happening in the wet lab and the computational part of doing genome assembly. From my background, I started off as a math major, and then in grad school I went to bioinformatics, but that's where I learned how to do all my wet lab work. And so I think having that interface between the wet lab and the dry lab is really important. So that's one of the motivations for this workshop. And so participants came and actually brought some of their isolates to sequence, and they did high molecular weight DNA extraction because you need long DNA to sequence, right? So if you don't have long enough DNA for the long read sequencing, you're not going to get long reads. So that's a really important part of it. And then we sequenced on a nanopore minion. And then we had the computational section actually using KBase to assemble these genomes. And then we, hopefully, I'm hoping participants will do this, but we also talked about how to do microbiology resource announcements, and Ben will talk about that. But if you're interested in the wet lab resources, they're on the website, and the slides from the workshop are also on the website too. So just like also kind of reiterating, so your sample preparation and your sequencing, how deep you sequence, your high molecular weight DNA extraction will impact your assembly. So this is something to keep in mind if you're interested in long read sequencing. And so let's see, well, I talked about this. So we had a lot of, because there was a wet lab and a dry lab section, we had a lot of TAs, and then actually getting everything into KBase. So thank PER for the funding, and Enigma and KBase for the support. There was a lot of work putting on this workshop. So just as part of the workshop, and this exercise is actually on the website as well, is I had people do this hands-on assembly exercise to really think about overlap. Like, what's the importance of having enough overlap with the reads? And I'll talk about that a little bit more, because if you want to run fly, you can actually specify the overlap. And people often like to reduce it to increase the contiguity of their genomes, but you will probably get misassemblies if you do that. So that was something I was trying to impress upon people. And then this is doing the high molecular weight DNA extraction, and we got sequencing from, these were pictures I took after an hour. So I think it's really kind of gratifying to do nanopore sequencing because you can see it right away instead of just sending it off to a facility and then getting the data back. So these are major concepts that you should keep in mind if you're doing genome assembly. So genome assembly is a guess, right? Like, we have these algorithms that try to put the pieces, which are reads, together. But depending on the algorithm you use, you may get a different assembly, which you'll see when I go over the narrative. The sequencing error rate will affect the assembly and actually the annotations, which I will also show some examples of. And the depth of sequencing also is super important. And I will also demonstrate this in the narrative. So these are all things of how the wet lab affects your assembly. These are slides from the workshop. I think we're going to skip that because I've kind of emphasized some of these points already. But, well, actually, I'll go over this. So basically, if you haven't thought about how assembly algorithms actually work, it's like you have a stack of newspapers. It's like many, many cells, right? You get a clonal culture, and then you extract the DNA, and then you end up with this pile of reads, but you only want to assemble one genome. And so that's actually basically how assembly algorithms work. Now, with long read sequencing, it's important to keep in mind the error rate compared to short read sequence. So there's like this difference with the length of the reads and also the error rate. So Illumina has about 1 error per 1,000 bases. So that's like an error per gene. But PacBio and Nanopore have an error rate that's much higher. So you might be saying, oh, I thought PacBio was supposed to be more accurate. So the only reason why PacBio is more accurate is because you can get hi-fi reads and it goes over, let's see if I have it here. So they attach these dumbbells to the side and then you go over the same read over and over again. And that's, you can get the errors out. So that's why it's actually, the raw data is actually pretty noisy. The other thing to keep in mind if you're doing long read sequencing is how much DNA you need. So that's the other limitation of long read sequencing is you need a lot more DNA than for the short read sequencing. The other comment I want to make about nanopore sequencing, so I would say when nanopore first came out, it had this reputation of having a really high errors, right? And it's because it's trying to, what you do is you feed the DNA through the pore, you get this current readout, and then you use machine learning models to figure out what the base is. And so because of DNA modifications, that actually alters the signal, so it's good and bad. You can detect DNA modifications, but you also get a higher error rate because you can't interpret the signal. So nanopores has been improving the models, and they just came out with a model that has that was trained on microbial data. So the error rate has gone way down. So if you think that nanopore sequencing is very inaccurate, the newer chemistries and the newer models, it's starting to get close to where PacBio is. Okay, so this is something I hammer home a lot is because again and again, I've seen people who just think they can send DNA off and then don't think about the depth of sequencing they have. But the coverage is so important. And it's because the coverage helps you remove errors from your genome. And so let's see, there's a little bit of error here. But basically, we can see that it should be an I, right? Because you stack up the reads, and then this is just one error, right? And so this is, it should be an I. So that's why the coverage helps with your genome assemblies. So coverage is really important. I can't really show you guys it really helps to do the hands-on exercise to think about overlap. But more overlap is obviously better. You can think of it as, would you rather have a puzzle piece that is bigger or tinier, right, for a whole picture? And then the length of the genome, I'll get to when I go over the narrative. So here's kind of just a visual representation of why repeats cause misassemblies, right? If you have too small of an overlap, then you can miss this whole green part of the genome. But then if you have long reads that start to span the repeats and you have enough overlap, then you'll get the correct result that you're looking for. Okay, so higher overlap is better. Longer reads help with overcoming repeats, and coverage is really important. So kind of a rule of thumb. So this is just through empirical experience, right? 50x to 200x coverage for Illumina-only genomes. And it's really good to get 50 to 100x for long read genomes. Okay. I can pause here. I'm going to start getting into the narrative if anybody has questions so far. Okay. So there's two types of, so when you're using long read sequencing, you could just do long read only. So that would just be using fly, which it's kind of like, yeah, so you would just use fly, and then there's a little bit of polishing and fly with long read only. You can also polish with Medaka, which is not in KBase. But if you want to do hybrid assembly, there's two ways to do it. You can do short read first. So you assemble all the short reads and then you thread the long reads through. So that's unicycler. And then you could do long read first, which is fly. And then you can polish with short reads because they're more accurate. Okay. This is just hammering home the wet lab stuff. You need high molecular weight DNA for the long reads. The other thing to keep in mind is, like, if you're looking at SNPs, you have to take a lot of time to polish your genome because the error rate's still a little bit higher than if you're doing short read only. Okay, so before I move on to the narrative, I just really want to thank everybody in the Enigma SFA and Torben and John Mark and Paul for this project, and then the support I've gotten from KBase, so especially Alicia, Ben, and Ellen. A lot of these apps are still in beta, so if you click on this little R here, if you can click on it and then you can see the beta apps, and I'll turn it to a B. Ben, I think I probably don't have to go over the structured narrative, right? Okay. Oh, you have a question? Oh, let's see. Okay, it should be public now. Yeah. The other thing that I wanted to mention is that there's a bug in the FASTQ upload. So you can't upload a FASTQ file if there's reads that are less than 10 bases. They're working on changing this. So this originally wasn't an issue if you had short read only, but with nanopore sequencing especially, there's a lot of reads that are short. You might want to just filter them out anyway, and you can do that with seqtk, and I put a example command here. But but this should be fixed. And then you can filter out the short reads with fit long. Okay. So I'm using this example of the Pseudomonas to show you. It's 9.4.1 data and Illumina data, so we can do a hybrid assembly and a long read only assembly. When some of the error is going to be exaggerated compared to what's now because they're on to 10.4.1 chemistry, but it's good so you guys can see the differences in the types of data. So with any type of with assembly, you want to quality filter the reads and trim off the adapters. And so for the Illumina data, I use Trim-O-Matic. And then for the nanopore reads, the base caller, which is currently Dorado, actually can trim off the adapters. So we're not doing it inside a K-base. And then I also filtered reads with fit long, and we'll use that a little bit later. So this is the long reads. So I'll be covering short read only assembly, long read only assembly, and then hybrid assembly, and then we'll compare how all of the assemblies look. Okay, so we did a short read only assembly with spades. And let's see. So this assembly had 33 contigs, and the largest contig was about 874,000. This is actually a pretty good assembly for a short read, a genome assembly. The other thing to notice, if you haven't done a lot of genome assemblies, is that the length is about 6.2 megabases, which is pretty standard for a Pseudomonas. So it's good to check if the taxa that your sequencing has had other genomes to see what other genomes look like. So if it's wildly different, then there's something probably wrong. And then also it's good to look at the N50 because that tells us how contiguous the genome is. Okay, so the next one in this narrative is long read assembly with fly. And so when you're assembling with fly, it's important to know what type of data you have. And let me just show you. So if fly can handle PacBio, and then the different types of nanopore reads. So since we're using 9.4, I'm using the less than 20% error. There's usually if you do fast base calling, you're doing real time base calling as you're sequencing. The next step is high accuracy, and then there's super high accuracy. So there's a little bit, so we can see here that the long read only assembly was actually pretty good. It has 2 contigs. Also note though that the length is a little different. So this goes back to genome assembly is kind of a guess. And then there is actually a little bit of a bug here. It's not showing you the coverage. So I put the result here. You can see it, you can see the correct file if you download the zip file, and then there's a assembly_info. And that's kind of the main fly output that you want to look at. So one thing to note about this assembly, which I think is important, especially if you're looking at legacy data, is that with nanopore sequencing, you can actually put in a control DNA sequence. And so it used to be lambda. And so this small circular piece in here that has really high coverage is actually the lambda phage genome because it was put in as a control. And then also, you note that this genome didn't circularize. So this is likely because the coverage wasn't high enough. It's only 15. Okay. So after you do your long read only assembly, um, it's good to polish it. So I polished it with the trimmed alumina reeds. And let's see. Okay, so in this one, so this is the summary. Um, so here's the main genome. So it actually changed 1% of the genome. So that's pretty high. Like I said, with the newer chemistry, this is not going to be as much, but it kind of gives you an idea of how inaccurate nanopore sequencing used to be. Okay, now with unicycler, so you input the trimmed alumina reeds, and then I'm inputting the filtered long reeds. So you could just put in the long reads by themselves, but the way that unicycler works is it does the short reads first, and then it tries to wind all of the long reads through the contigs. And so if you have too much long read data, it might take too long to do the assembly. So that's actually one reason to filter. One reason I didn't use the filtered reads, I forgot to mention for fly, is because it actually filters out all of the reads that are shorter than the specified overlap. And so you don't have to necessarily do that. So unicycler, we got, yay, one genome, and it's circular. So that ended up working out really well. So note that this genome's actually a little bit closer to the spades assembly in length. This is actually the output that you get if you're running unicycler on the command line. So if you want to learn more about assembly graphs and everything, that's what's going on here. But you can pretty much see all of the results in the summary that we have here. Okay. So I was going to make this a little bit interactive, but I think because we're of time, I'm going to keep on going. Basically, this first question, what do you think about the length differences between the different assemblies? It's because the algorithms are different. Why didn't the lambda fade show up in the unicycler assembly? It's because it's short read first, so it didn't have any lambda contigs to wind the long reads through. I covered this third point because it's because of the coverage wasn't high enough. And then these two questions actually apply to what I'm showing down here. So one place to see the effect of the errors is to actually look at the number of genes and the number of hypothetical genes. So we can kind of use unicycler as the gold standard here. And if you expect about one gene per 1,000 bases, so this looks about right for this genome. So there's about 5,500 protein coding genes in this assembly. But if you look at the unpolished long read only assembly, has 12,000 genes. So the errors actually caused miscalls on the genes. And then if you polish the genome, you actually get back to much closer what you're expecting. If you type in, let's see. So if you go to the output and you look at the hypothetical genes, you'll actually get way more hypothetical genes as well here compared to the polished one. And it's just because the genes are too short, you can't, the algorithms can't figure out what the genes are. So even though there's like twice as many genes, a lot more of them are being called as hypothetical in this unpolished genome. Okay. So one way to learn a little bit more about how coverage is affecting your assembly is to use this Circos tool and to look at the number of contigs you're getting. So this is the original short read assembly with 33 contigs, and it had a coverage of about 122, which is that's pretty good. You can aim for at least 100x coverage. But if you subsample, so the coverage is lower, so you can subsample reads in K-base, then you end up, so if you look at the spades assembly, if you subsample, so I subsampled the 10% of the reads, which is extreme, but you can kind of see what's going on. You get 438 contigs. And so the coverage was only 13. And so you can see that it just ended up in a much more fragmented assembly. So it's just one thing to keep in mind as you're doing your sequencing. And then one thing, right? So even though the coverage really affects the assembly, if we still did the hybrid assembly with the subsampled reads, the long reads are adding a little bit more coverage, but you could actually still get a pretty good assembly with less data or less sequencing if you have the long reads added to it. If you get into looking at assembly graphs and you want to analyze these assemblies, you can use something called bandage. And I don't think I have enough time to kind of go through this, but it visualizes the assembly graph and it'll look actually something like this logo. And so that's using all of the apps that we put in.
All right, so thanks, Lauren, for that presentation about some of the new long read assembly tools and the workshop. Just quickly, before we end our workshop today, what I wanted to share with you all is some of our vision for K-Base as far as publishing and sharing data that users are generating. So like Lauren showed, we have a lot of tools for processing reads, assembling genomes, annotating them, and so on. But one of the really unique features of K-Base, especially compared with some of the other platforms that are out there, is that we have sharing and collaboration features that allow researchers to work together, and also for people creating data on the platform to publish that data independent of their publications, but including that data in their publications as well. So again, one of the things that makes K-Base very unique is that we can work together on the platform. So if you've, for instance, assembled and annotated a genome and you want to share it with your colleagues to analyze it together, you can do that within the narratives, and I'll show you how to do that in a bit. But you can also share your work in your publications for the larger community to have access to as well. So this just kind of demonstrates that after assembly and annotation, there's quite a few further analysis that you can do on the system using all of our apps and tools. Another way that you can share things on the platform is via organizations. So organizations are a unique feature of KBase that allow research groups, labs, whole institutions to uh, collect their narratives, collect their data, and pull them into a organization where everybody inside of that group can have access to each other's narratives, to their data, and work together directly within the narrative notebooks. And so we see not only Department of Energy groups like Enigma or projects like Grow using this, we see lots of labs at different universities or educators using this to organize their training materials and classroom materials for bioinformatics and genomics. So it's a great way to give everybody in a group access to all of the data and analysis that they're working on together. So part of the vision of KBase is this sort of social feature where people are putting their data out there into the world, and other researchers are finding that data in the publications or on system and doing their own unique analysis. So this graphic just shows the example of two different researchers sharing their narratives with each other and analyzing it in the context of public data. I'll also show you some of the ways that KBase is incorporating large data sets from different groups. But one of the things that really excites us is that we're seeing organic use of these features from our user community. This slide just shows you a couple of different publications that have come out within the last year or so, where users are sharing their narratives in their publication. So as anyone is reading their publication and they want to see how the data was processed, they can go directly into the narrative and find the data, load it into their own narratives, run their own analyses and comparisons, and even feed that back to the original researcher. And one of the really cool things about this is that we can mint a DOI for narratives so that the actual analysis on KBase has its own unique identifier beyond the identifier for the publication. So we've seen examples where users will have a single narrative that's shared between multiple publications that has its own DOI, so that they can treat that as a research product that they've created. So we really built some of these features around the concept of FAIR, that's findable, accessible, interoperable, and reusable for both the data products and the analysis that are going into the system. So we have a feature called static narratives or FAIR narratives that basically creates a HTML snapshot of the current state of a narrative that creates a URL where anybody, regardless if they have a KBase account, can access that URL and see the state of the analysis and data as it was generated. That's also very useful because the researcher who initially created the narrative may want to continue to analyze their data as new tools come out on the system. So the snapshot remains persistent, but their actual narrative, they can continue to add and change and continue doing their research. If you have narratives that you want to include in your publication and create a DOI for, right now this is a manual process where you can just reach out to us and we will help create your DOI for you. But it's useful because it's indexed by search engines, so it helps kind of push your work across the internet. So you're not necessarily only relying on people accessing your publication to find your work. And this just kind of shows you some information about how much our system has grown, how much science users are doing on the system, and so on. But I just want to go and share with you all a couple of these resources that I've mentioned. One is this MRA genome announcement template. So we get quite a few users publishing genomes and microbiology resource announcements. This narrative helps you prepare your data for publication, and has just helpful things like check boxes for each step so that you know that your data is ready to share. So I just want to show an example of a user doing this. This is from some of my colleagues at Oak Ridge, where they have sequenced a organism on the roots of Populus deltoides. So if I go down here in their publication, you can see that uh, they've shared a link to their narrative here. So if I click that, what it will show me is the static narrative that they've created. And again, this URL is accessible by anybody, regardless if they have a KBase account or not. But what I can see is this rendering of their narrative with all of the data and analysis that a random researcher can explore. But it also gives you the ability to see their data and copy that into your own narrative. So for instance, if I click their genome, it will open up a page for their genome with all of the information about this particular organism. I can look at provenance to understand any processing that's been done on this piece of data, any of the tools or settings that were used in creating that. But again, the nice feature is I can just click this copy button, select the narrative that I want to, or create a new one. It says it's successfully copied that. If I click open my narrative now, it should load for me the narrative with this data loaded in it. So just, in just a couple of seconds, I'm able to access their data and start doing my own analyses on it if I wanted to, find apps that I can process this or so on. And so you can see that this is a very rapid way to start building out and sharing your analysis in a collaborative way, and allows for continued use of all of the genomes that are being produced today, because as we know, there's thousands and thousands and thousands of new microorganism genomes that come online all the time. We hope that this will actually enable the research community to dive deeper into what's inside of those genomes. Just a few more things. So just to show you how you can create a static narrative, since I just showed an example of users doing that. So if you're in the KBase narrative interface and you go to the top right where there's this share button, first you want to make sure that your narrative is public. So I'll shift this to being a public narrative. And this is just an example, but then I go down to where it says manage static narratives, and you can see I can create a static narrative, and this will create this link that I can use. There's nothing in this one except this genome, but you can see how easy it is to build a shareable narrative that you can share with your colleagues, share in your publications, and so on. And one of the last things I just want to show is that as an example of how we are trying to increase these sort of sharing and collaborative environment in KBase. So one, I had mentioned organizations. If you're in the main navigator page and you go to orgs on the side here, you'll see all of these organizations that our users and our research community are building here, including, if you type in Lisa, you can access the materials that we've shown here already. But just again, to show you kind of the future of what we're looking to do, we just put this out, I think, last Thursday, so very excited to show you all this, is that we're releasing whole collections of data that affiliated groups are putting together. So for instance, the Enigma project that Lauren works on, you can go here and access all of the genomes in their collection and very quickly start to select. You can also search your own genome against this collection to identify hits based on homology, put them in your own narrative, do your own analysis. So we really hope that the platform will continue to attract users who are interested in sharing their work, sharing their data, and doing collaborative analysis together using the unique tools and features that we've put together. So thank you all for coming, and hope you have a good rest of your GSP meeting.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.